of you guys have not done your quiz yet? Okay. You have until 6 to do it. What happens at 6:01? It is closed and you get a 0. So it doesn't even take that long, guys. Okay. That last question, question 14, you've never done a click on target before. Why did I give you 5 chances? Because I didn't want your spazzed out finger to be the one that it counted, right? So there's really only 2 things to click on, but I gave you 5 chances. Does that make sense? You can do that, sure. It takes the last 2 clicks that you have, or you can undo a click, right? You can undo it. There's only 2 things to click on, okay? FYI. So, but people get spazzed out and they're like, oh, I just touched it. And so it doesn't matter, huh?
I was trying to label all the vesicles.
There's only 2 vesicles. The other 2, after they leave, they're no longer a vesicle, right? That makes sense?
Yeah.
Yeah? Okay. So there's only 2 vesicles there.
Last 2 were those ones down there.
No, but they're not a vesicle anymore because they've actually already joined with the cell membrane.
Okay.
Right? Does that make sense? The vesicle part's kind of gone?
In the vesicle
outside.
Right. Well, the vesicle just joined with the cell membrane. It's no longer a vesicle. Got it? Okay. So you don't have to click anything 5 times. It just says 2 out of 5 because, you could have clicked 5 times, right? That's all. I was just trying to give you a little wiggle room. Almost everybody got it right anyway, right? So you do not need to click 5 times. And you can still go back and fix it up until 6, right? And undo and whatever. Yeah, you can clear the whole quiz out and start over if you want. It doesn't make any difference. So you should get 100 is all I'm saying, right? If you get a 0, sucks to be you. Okay, you ready? We're going to finish synapses, and then we're going to start on muscle today. We did endocannabinoids in here, right?
Yeah.
Did we? Okay. All right, so the next thing that we're going to talk about, all right, it's going to be dry. Not a damn thing I can do about it, but you're going to have to stick with me. So first thing I want to talk about is we're going to switch from talking about the neurotransmitters to talking about the receptors. The neurotransmitters are more interesting to talk about than the receptors, but can we all acknowledge that the fact that it's really the receptors that cause the cell to behave in whatever way it does, right? Because acetylcholine could have an activity on one cell which was excitatory and be inhibitory on the other cell. So receptors are super duper important. In addition, how much clinical research do you think goes into receptor function? Like drug research is mostly receptor and reuptake mechanism function, right? I mean, of course you have to know what the neurotransmitter looks like, but it doesn't do you a darn bit of good unless you know how it behaves at a specific cell by looking at the receptors. So all I'm going to do is I'm going to teach you a little bit about receptors, a little bit about functionality. You will run into this stuff again when you do pharmacology, if you're doing nursing, right? Or if you're going into pharmacy and you hate this stuff, change your career path, all right? Okay? Because it's a whole bunch of receptor conversations. All right, so first, go ahead.
Quick question about the quiz. So
Yes.
Is that the level of understanding, or is that just enough for the test?
That's the first level of understanding. That is the first time you see neurotransmitters, I need you to know it at that level, right? But it's going to get a little deeper. So you've probably noticed that the quizzes get a little deeper as you go from the first quiz to the second quiz over the material, right? So when we have our next quiz next Wednesday, it'll have deeper neurotransmitter questions and shallower muscle questions because you will have just started muscle. Does that make sense to you? Okay, so that's kind of what I'm trying to do with you guys. Like the first exposure is like, have you organized your information? And the second time is like, can you apply it, right? That makes sense? So, but a lot of people have trouble just with the organization step. They're like, you just threw a bunch of terms to me and I have no idea how they fit with other terms. And that's what I wrote that one for. I'm like, can you get all this stuff organized in your brain, right? All right, so let's talk about receptor naming before we get into the specific receptors. You have noticed that some receptors are named for the neurotransmitters, like GABAergic, but some of them are named for a drug, opioid receptors. Why is that? It's just because of history, really. It's just because sometimes we discovered the drug before we even knew anything about neurotransmitters at all or anything specific about the neurotransmitter system. Opioids are a great example, right? How long were people doing opium? A hell of a lot longer than we've been studying neurotransmitters. Yes? So we called the receptors opioid receptors, and then later on we discovered endorphins and enkephalins. Okay? Does that make sense? Okay, so, however, see if this logic makes sense to you. Do we actually have opium receptors in the brain, or do we have receptors that bind to an endogenous chemical that also bind to opium? The chemical had to predate the drug interaction. Otherwise, when we smoked or licked or whatever that drug, it would have been a non-starter. You'd just be like, meh, probably won't smoke that plant again. So the endogenous chemical always predates the drug. And you could argue that the opium poppy and cannabis are actually just using us to make sure that their evolution continues. Yes? And it's pretty damn effective, isn't it? Like, we don't have any risk of cannabis actually dying out as long as human populations are on this planet, right? So they're just hitchhiking a ride onto our evolution, and it's a pretty effective mechanism, you think? So, but the endogenous chemical predated the drug. Otherwise, there would be no reason to go and seek out the drug. Yes? Because it's actually working with the chemical system. Go ahead.
What is endogenous?
Oh, endogenous means that you make it.
Oh.
Yeah?
Okay.
So your endogenous chemicals are chemicals that you make naturally. Does that make sense to you guys? So it may be a semantics question, but it's also an interesting logic thing that you guys should be able to figure out. So depending on the history, either the drug gave rise to the receptor name or the actual neurotransmitter gave rise to the receptor name. So with opioids and cannabinoids, right, we learned about the drug first, right? But with GABA, we knew about GABA before we knew about benzodiazepines. Does that make sense? Otherwise, they might be called benzodiazepinergic, which would suck, right? Okay, so they're called GABAergic. Everybody good with that? Okay, so now what we are going to do is we are going to learn signal transduction pathways. This stuff really seems a little hard to make sense to students, but you're going to have to learn it. FYI, this particular little part is usually what people do the worst at on the next test. And I think it's because it's really hard to make it relevant and interesting. So I'm going to do my damnedest, but you have to engage in the process with me. Ready? Okay. So what we're talking about now is what happens after a chemical messenger binds to a receptor. Okay? What happens inside the cell to actually cause the cell to behave differently. Obviously, the cell is going to behave differently after a chemical messenger binds. Otherwise, why the hell bother, right? It's going to do something different. It's going to have a new protein, or it's going to depolarize or hyperpolarize, so it's going to do something. Okay, the process of getting the target cell to respond to the chemical messenger is called a signal transduction pathway. So, here it is. I'm a signal. I'm a neurotransmitter, perhaps. She's the receptor. This is the cell membrane. You guys are all of the organelles and enzymes inside the cell that could cause things to happen. Okay? If I am a water-soluble chemical messenger, then do I get to go inside? No, I don't, right? Okay? That means if I want something to happen inside the cell, we have to play telephone. I bind to her, she turns around and communicates to some enzyme or organelle, right? And then ultimately, I want to get a message all the way back to Uinta, right? Her, okay? And I have to stay out here. So what we have to do is we have to play telephone inside the cell. So it's a signal, and you have to transduce the signal, right, to get it to there. Maybe we want protein synthesis to occur, and we need to get the signal all the way to the nucleus, which has a specific gene that we're interested in, right? That's a long game of telephone. That makes sense? So I have to stay here, she has to stay here, we have to cause something to happen. It's called a signal transduction pathway. However, if I were a lipid-soluble chemical messenger, couldn't I bypass all of you, right? I could go straight to a receptor, which may be in the nucleus or in the cytoplasm near the nucleus, and go all the way and activate the gene directly. Is that a longer or shorter game of telephone? Much shorter game of telephone. Does that make sense? But either way, these are called the signal transduction pathways. And a signal transduction pathway is how you get the target cell to respond to a chemical messenger. Got it? Okay. So, we are going to do the easy one first. The shortest game of telephone uses blank soluble chemical messengers, lipid soluble chemical messengers, and where am I going to find the receptor? On the cell membrane or intracellular? Intracellular receptors. You guys with me? So I'm going to use a lipid-soluble chemical messenger, I'm going to use an intracellular receptor, and I'm going to bypass all of the telephone that I can, okay? So this process is going to be called gene activation, because watch what it's going to do, okay? So this is some lipid-soluble chemical messenger. It goes straight through the cell membrane, and it finds its receptor either in the cytoplasm, or it can find it all the way in the nucleus. There are actually receptors that are in the nucleus. Regardless, once the chemical messenger binds to the receptor, that's a new shape that didn't exist before. The receptor chemical messenger complex is a new shape. That new shape will now go directly to a gene, specifically before a gene, and cause transcription, right? And then what's going to happen after transcription? This goes out the nuclear pore and does translation. And now ultimately what I have is a new protein. It could be a new structural protein that changed something structural about the cell. It could be a new enzyme that changes something functional about the cell. But notice that I skipped all of the intermediaries that I could, right? Okay, let me show you an animation that will make this make sense to you. Okay, you ready? Okay. This is aldosterone. It just happens to be a hormone. It doesn't really matter what kind of chemical messenger it is. It is lipid soluble. Goes straight through the plasma membrane, finds a receptor, goes through the nuclear pore. This goes straight on to find something usually upstream of a gene, forces transcription. That goes right back out the nuclear pore, headed to what organelle? What organelle? Ribosome, exactly. Does translation, gets me a new protein. What is that protein? I don't know. It depends on what cell you're talking about. But it's a new protein that can change either the structure or the function or both of a cell. Does that make sense? Okay. So if it's a new enzyme in a metabolic pathway, then the cell behaves differently, right? If it's a new structural protein, you might change something or structural about the cell, okay? So, but regardless, what I did is I forced some kind of protein synthesis in the cell directly. So, this is called gene activation. Examples that we will run into in the future and reasons you would care about this as someone who's going into clinical anything. Estrogen receptors work like this. Cortisol receptors work like this. So you know that we actually treat estrogen disorders quite often, and we need to have things that we know will bind to estrogen receptors, right? So, and if we make an estrogen analog that's similar to estrogen, it actually has to bind to the receptor, right? Cortisol is a hormone that I don't know if you guys know about. It's one of the hormones that you absolutely need to keep you alive, and there's lots and lots of research into cortisol receptors, okay? So what these do is, these are obviously lipid-soluble hormones. They go straight through the cell membrane, and they actually cause this to occur. Everybody good? Okay, all right. There are other ways that lipid-soluble chemical messengers can work, but this is the most common way, okay? All right, so now we get into the ones that there are 4 of these, and they're a little harder to understand, but we can do it, okay? So these are chemical messengers that are going to bind to plasma membrane receptors, which means that they are going to be blank soluble chemical messengers, water soluble chemical messengers. There are far more water soluble chemical messengers than there are lipid soluble chemical messengers. Okay, so there's going to be more of these. We're only going to learn 4. We're going to learn the 4 that you're going to run into this semester and in the future. You might run into more of these. Actually, you already know the first one though. So, water-soluble chemical messengers attach to cell membrane receptors. And are they going to play longer or shorter games of telephone? Much longer games of telephone. Some of them are just silly long, right? I don't even know how it evolved that way. But the first one isn't very long. It's still longer than going directly in there, okay? So you've met these before. This one has a chemical messenger binding to a ligand-gated ion channel or an ionotropic receptor. You've met those before, right? Yeah, ionotropic receptors. Let me show you a picture so that you can remember that you've met them before. Here we go. Chemical messenger binds to ion channel, opens it, or you could close it. Either way, what you're doing is allowing ions in and out of the cell that would not otherwise be allowed in and out of the cell, right? And the most common effect of that is that you can depolarize or hyperpolarize the cell, which is of course what neurotransmitters do, right? But that could not be the point. There's another reason that you could do this, just FYI. Just so you know, you're not always trying to cause depolarization or hyperpolarization, even though it will occur. Sometimes what you're trying to do is let calcium, for instance, into the cell, because calcium is going to result ultimately in protein synthesis by a long game of telephone. Calcium binds to something, activates it, starts a metabolic process, goes all the way to the nucleus, and causes protein synthesis. It's going to be a much longer way of doing the same thing than gene activation. Or calcium could enter the cell, and instead of just depolarizing, which it would, it could also cause muscle contraction, or calcium very often is a mechanism for causing secretion. You actually know of an instance in which calcium moving in causes secretion, right? Right? In axon terminals, right? It's not ligand gated, but you know of an example in which calcium moving in causes secretion. You with me? So basically, in this particular situation, what I'm doing is opening or closing an ion channel, and something intracellular changes because I did that, right? I changed the composition of the intracellular fluid, which is the trigger that changes the cell function. Got it? Okay, so why do you care about these? You care about these. First of all, we're going to see calcium channels really common. Calcium is really high in the extracellular fluid. You can use it to cause exocytosis and all kinds of things. Second, nicotinic cholinergic receptors are these kind of ionotropic or ligand-gated ion channels. Are they going to be fast or slow to cause their responses? No, they're pretty fast, right? Because what these do is immediately let the ion in, and the depolarization can get you an action potential. Now, gene activation is even slower than this one because protein synthesis is slower than just, for instance, an action potential. So these are going to be the fastest of the cell membrane receptors. Okay, so these are called ionotropic receptors. Why do we care about these? Nicotinic cholinergic receptors are the ones that we use in the central nervous system for learning, attention, and memory. We are studying these because these are the ones that degenerate in Alzheimer's disease. So these end up being important clinically. Does that make sense? Okay, all right. They're also, and part of the reason we know so much about them is that they are also located in the peripheral nervous system. You have nicotinic cholinergic receptors on every single skeletal muscle cell that you have. So we have lots of opportunities to study them, right? Because you can actually stimulate this and block it and paralyze the cell without killing the person, right? You can even do living experiments on a person and it won't even cause permanent paralysis. So we have lots and lots of research on these nicotinic cholinergic receptors, which are ligand-gated ion channels. All right, next one. Again, it's going to be a cell membrane receptor, so it's going to act with blank soluble chemical messengers. Water-soluble chemical messengers. Okay, here's the next one. The next one is called a tyrosine kinase receptor. So stick with me for a second. This time, instead of the chemical messenger binding to and opening an ion channel, the chemical messenger is going to bind to and activate an enzyme. Okay? And notice that the enzyme part is faced toward the intracellular fluid, which means you can change the activity of the cell. Okay? So what happens is the chemical messenger binds, yeah? And it activates this enzyme, which was sitting around in an inactive state. And it activates this enzyme, and what this enzyme actually does is it causes phosphorylation. What's phosphorylation? Yeah, adding a phosphate, because it's a kinase, right? Causes phosphorylation of the tyrosine amino acid of another protein. Okay? Why the hell do you care? Phosphorylation is a really common way to activate or deactivate a protein. So I had a protein that was sitting around not doing anything, phosphorylated it, and now it's active. So now the cell behaves differently than it did before, right? So these are called tyrosine kinase receptors. What happens after that? Well, the cell does something that it wasn't doing a minute ago. It activates some metabolic process, it turns on protein synthesis, it turns on growth, it turns on something, right? Okay, so these are called tyrosine kinase receptors. In this instance, the receptor is not an ion channel, the receptor is also an enzyme. Okay, got it? Why the hell do you care about tyrosine kinase receptors? Well, you might not, but we will run into tyrosine kinase receptors as a very common class of hormone receptors. And we sometimes have hormone problems that need to be treated by knowing exactly what the receptor is supposed to look like for a hormone. Okay? All right. So next one is just like that, only with one little difference. Okay. The next one is called, and your textbook doesn't have any pictures of it, which is bizarre because they're actually the most common of all of them. These are called the JAK kinases. So just like tyrosine kinase, except one difference. You ready? The only difference is this receptor part is a separate protein than this enzyme part. With the JAK kinase, they're all part of the same protein. I'm sorry, with the tyrosine kinase, they're all part of the same protein. With the JAK kinase, there is a receptor protein that's made by a different protein synthesis technique than, well, not the technique, but a different protein synthesis process than the kinase. Regardless, still what it does is it's a kinase, so it phosphorylates an intracellular protein and starts the activity, right? Okay, so why do you have to know about these? They are the most ubiquitous receptors that we have. They're so damn ubiquitous that JAK kinase actually stands for just another kinase. Because there's so many of them that they got tired of naming them after themselves, Jeffrey Sorensen's kinase, and it's just JAK 254. They don't even care when you find one anymore. You're like a biochemist who found a JAK kinase, and they're like, oh great, did you find hay in the barn too? So there are so many of these, okay? But the only difference between this and a tyrosine kinase is this is a separate protein than this, okay? So these are really, really common, the JAK kinase. And why will you care? Well, growth hormone, which is important, of course, for growth, but also for cell division, for protein synthesis, and for repair of wounds, interacts with JAK kinase receptors. Prolactin, which is prolactation and is involved in the whole nursing process, binds to a JAK kinase receptor. So you're going to learn about these proteins a little bit later. All right, and now the last one. This one, I seriously cannot fathom how this evolved because it's so damn unnecessarily complicated. But it's the last one. Ready? These are called Gprotein coupled receptors, and they are almost as common as JAK kinase receptors. And they are just doing it by the most circuitous route possible. So, whereas you could, with this guy, you could bind directly to an ion channel and open or close it directly. This is how Gprotein coupled receptors do it. The Gprotein coupled receptor will use 4 different proteins to activate the 5th protein. So this is like, instead of shooting straight for the pocket when you're playing pool, this is like doing a 5-ball combo shot to get in the same pocket. So look, here's what happens, and I have an animation for you too. So this is the ion channel that we're going to open, right? And we're going to change what ions are in there. But instead of binding to the ion channel to open, we're going to bind over here. And then these 3 proteins that are activated by GTP, that's why they're called G proteins, these 3 proteins will slide through the cell membrane until ultimately one of them will smash into that ion channel and open it. It's ridiculously complicated. And I can't fathom how this is so common because it looks like there's so much that could go wrong, right? So, and it could get even more complicated than that. Sometimes G proteins do not even directly smash into the ion channel that they will open. Sometimes they do it this way. So I'm not going to hold you responsible for this, but look at this. Bind to here. Ultimately what I'm trying to do is to open that ion channel. I bind to here, slide these 3 through, activate the enzyme, which then makes something that opens the ion channel. What the hell, right? What do we call this in Texas? We call this going around your ass to get to your elbow, right? It's the most inappropriate, like needlessly complicated way that you can possibly think of to do something. But apparently it worked because there's tons of these. So I'll show you an animation that shows you how these work. You ready? So Gprotein receptors. So ultimately what I'm trying to do is just smash into this ion channel and open it. But let's see how you get it going, okay? All right. So ultimately I have a cell membrane receptor, okay? And this guy is water soluble, right? So what it's going to do is bind. And when it binds, it changes the shape of the receptor. Please note that GTP, which is guanosine triphosphate, is used as the energy source. That's the only reason they're called Gprotein receptors, right? Okay, you don't have to know how it's used, but notice that alpha, beta, and whatever the hell that is, are sliding through the phospholipid bilayer. Ultimately, what's going to happen is alpha is going to get loose from its friends, And alpha is going to slide over and smash into that ion channel and open it. Is this going to be faster or slower than directly opening the ion channel? Hell of a lot slower. Slower than Christmas, okay? And then what's going to happen is you're going to have to be able to use it again, right? So you're going to use GTP to actually put it back in a position till the next time that you use it. What? I know. So why am I making you learn this? Because these are so common that you're going to run into them as soon as you start looking at pharmacology. You're going to run into these. Go ahead.
Is there multiple benefits that could come from the products of, say, a Gprotein, other than just smashing into something? Because you said the longest one,
the longest one right here.
It says
it can do multiple different things. Other cell responses, or opens or closes. So it can do multiple things.
So is it just the aspect that it can just do multiple things in a row?
But it doesn't always do multiple things, right? Sometimes it just does that one thing, and then you're like, well, why didn't you do it directly? I don't know. Maybe you want the response to be really slow. I don't know. I've never really heard an answer that makes any sense to me. But let's see why you would care about Gprotein receptors, because you are going to care about Gprotein receptors, because muscarinic cholinergic receptors, which are CNS cholinergic receptors that bind to muscarine and acetylcholine, these are Gprotein receptors. They're going to be slower than nicotinic cholinergic receptors to respond, but they're still going to be important. Alpha adrenergic and beta adrenergic receptors. So alpha adrenergic receptors are Gprotein receptors in smooth muscle that when you bind to them, it will cause vasoconstriction, right? And that's important for maintaining blood pressure, right? And specifically, it's vasoconstriction going to your skin and your viscera. So for instance, when you get a sympathetic response and you become white as a sheet, that's those, okay? And then the reason it's also going to be important to you is you've probably heard of beta blockers before. Have you heard of beta blockers? Okay. So beta blockers are found as a medication that works with these receptors. It specifically is an antagonist for these receptors. So you've got beta 1 and beta 2 receptors. The receptors that are on the heart are excitatory adrenergic receptors. So when I get spazzed out and I release adrenaline or epinephrine or norepinephrine, it binds to the heart and increases my heart rate. But if I have heart disease, do I want my heart rate to go up? No. So what I use is a beta blocker, that blocks this receptor on the heart. And you use that as blood pressure medication, right? And because you're an American, you're probably going to end up with beta blockers with you or somebody you know, because we eat too much and we do not wiggle enough, right? So heart disease is really, really common in the United States. So we're going to run into beta adrenergic receptors. And then last thing, why the hell would you care about this really complicated receptor? Your serotonin receptors, which you do care about if you care about anything, right? Your serotonin receptors are Gprotein coupled receptors, right? And those just do every damn thing in the brain, right? Mood, sex drive, hunger, sleep, all of that kind of stuff. Good? Okay, so we're done. Done with the hard part, okay? So now I can tell you the interesting part. So the interesting part is, why did you do any of this? Why do you care about signal transduction mechanisms? Because ultimately you're going to change something about the cell activity, right? You're going to make it transcribe and translate a new protein so that you can behave differently. You can activate or deactivate an enzyme that was already there. You can induce secretion or cell division or depolarize or hyperpolarize, or you can change the metabolic rate of the cell. What does it do? Well, it's really cell dependent. So you wouldn't have to memorize it. You'd have to look at an individual cell and say, oh, I can see what it's doing in that cell. Okay? Okay, so note to self, because this is dry as a bone. This part right here is usually what people do the worst on the second test. They don't want to learn it. I didn't want to learn it either. I didn't even want to teach it. But you do not want to miss 4 questions because you would not learn the unpleasant signal transduction mechanisms, right? You don't want to do that to yourself. So just get it out of the way, pretend it's castor oil, take your medicine, get it out of the way. Okay, so let's review before we move on. You ready? Okay. If I am a water-soluble chemical messenger, I bind to a receptor where? Cell membrane. The fastest response that I can have with a cell membrane receptor would be to bind to a receptor that was also a what? Ion channel receptor, right? Okay. Those are called Gprotein receptors? Ionotropic. The rest of them are metabotropic, by the way, and they're slower. All of the rest of these are metabotropic. Okay, so the next way that I could do it is to bind to a receptor that was also an enzyme. Those receptors are called what? Tyrosine kinase receptors. Or I could bind to a receptor that was bound to its friend that was a separate enzyme, and those are JAK kinase receptors. Or I could decide to go around my ass to get to my elbow and do the most circuitous route possible to cause a actual cell response, and that type of receptors are called what? Gprotein receptors. Got it? Okay, so now let us move on and talk about one last thing before we move into muscles. I want to talk about how your nervous system can be responsive to some stimuli and not as responsive to others. Okay? So for instance, right now, you are sending relatively frequent action potentials from the sound of my voice, but you are sending relatively infrequent action potentials from feeling your clothes on your body. So why? How? How do you send different frequencies of action potential depending on the stimuli? Okay? So you can decide right now to attend to the feeling of the clothes on your body, and it'll increase the frequency of the action potentials. So how is that going on? Like, what is going on? Because action potentials are expensive, right? And some stimuli are worth more to you than other stimuli, right? So how do you change that? So it all starts out with the same story, no matter what. We know that an action potential can move about 250 miles an hour down like a myelinated axon, but then it's going to slow down at the synapses, right? That's called synaptic delay, right? But you always start the story the same way, regardless. You're always going to start with a local or graded potential, yeah? And if the local or graded potential is depolarizing and strong enough to hit threshold, then you'll get an action potential. But then we know in a cell, there are no strong or weak action potentials. Yeah, no strong or weak action potentials. So in a cell, an action potential is like every other action potential. That's called the all or none principle of action potentials. But we do know that the magnitude or the intensity of a stimulus can be communicated via what? Frequency of action potential. So here is the thing that I want to talk about. How can you have different frequencies of action potentials to different types of stimuli, and how do you decide? Most of the time you don't get to decide. Well, this is called adaptation or accommodation. And so basically what happens is you are going to, if you have a constant stimulus tends to get you a relatively infrequent action potential, but new stimuli tend to get you frequent action potentials. So what's going to happen right now is the clothes on my body are a relatively constant stimulus. Am I sending in zero action potentials about them? No. But what happens is, over time, with a constant stimulus, this stimulus is not threatening to me, right? It's not going to kill me. I can't eat it, and I can't have sex with it, right? If it were in any of those three categories, you would be predisposed to give it relatively frequent action potentials, right? But if it doesn't fit into any of those, after it's a constant stimulus, you can desensitize the receptors to that stimulus, and you can decrease the frequency of action potentials to that stimulus until it changes, like all of a sudden my shirt flew up, or I decide consciously to attend to it. So if you decide consciously right now to attend to the feeling of clothes on your body, then you can increase the frequency of action potentials, right? But most of the time you don't get to consciously decide to attend to things. Most of the time what you are set up for is you notice changes and you send very frequent action potentials for new stimuli. Okay, so let's talk about this. Even if you're not a parent, imagine for a moment, even if it makes you miserable, that you are a parent, okay? I want you to be sitting in one room and studying, and your children are in another room, and they're doing kid stuff. They're snacky, yelling at each other, playing a video game, knocking some crap over, right? Okay, so you're sitting in here, and you're studying, and you're attending to your physiology notes, right? And the action potentials are going in relatively frequently, and you're looking at your physiology notes. And are you not hearing that? No, you're hearing it, but it's a relatively constant stimulus, so it's going in relatively infrequently, right? And then bookcase falls over, crash. What happens to the frequency of action potentials? It goes in really dramatically, right? But does it have to be loud, or does it just have to be different? For those of us who are parents, sit in the room, and now you hear silence. Anybody who's a parent in here know what silence means? They escaped from the asylum, right? They are murdering one another, or they are in your crap. That's what it means, right? Ooh, digging through mom's stuff is what they're doing. So that's a really frequent action potential, but it doesn't have to be loud. It just has to be different, right? And you perk up because it's different. So why? Well, evolutionarily speaking, can you see the advantage to paying frequent action potentials and paying a lot of ATP to new stimuli? I want you to think about walking across the African savanna, right? I'm walking across the African savanna. I'm a proto-human, right? And I'm hearing a bunch of things. I got basically the babbling brook and the wind through the rushes and the birds tweeting, right? All of that's going in, and it's relatively constant, so it's infrequent, right? And then I hear a very soft, what happens? That needs to go in very, very frequently because it's a new stimulus, right? That makes sense? However, if I were a lion tamer living next to the lion cage, would it really? No, right? Because it's a constant stimulus, right? So basically, we are set up to notice changes and to devote relatively frequent action potentials to changes in stimuli. So have you ever driven through a town that's just like the entire town stinks? Like, yeah, so what?
Bozeman, Oregon?
Yeah, is it a paper mill town?
No, it's a coal plant.
Oh, coal plants.
A bunch of
In
different-
Texas and around Texas, there are tons of paper mill towns, and for whatever the hell reason, paper mill towns smell like butt foot. They smell atrocious. They smell like somebody just wiped their butt and then stuck the sock on their foot and wore it for 5 days, and then just walking around with it. So you drive through this town, and you're like, what the hell, how can you live here, right? How can they live there? Have they burned out their nasal epithelium? No, they have not. Can they still smell fart?
Yes.
Totally. Can they still smell perfume? Absolutely. But this constant stimulus just does not elicit very frequent action potentials any longer. Does that make sense? But you drive through the butt-foot town, and what do you smell? You're like, oh, what the hell is happening here, right? So you basically are modifying your input based on whether it's constant or whether it's new, okay? And the interesting thing is, because of 100,000, 200,000 years of human evolution, we do see some differences in males and females with how good they are in adapting or accommodating out extraneous information. And we haven't had long enough for us to change yet. So only for about the past 100 years have there been a lot, maybe 150, has there been a lot of overlap between male and female roles societally. And now you guys can totally have the same kind of jobs and stuff, but it's actually relatively new. And if you think back to hunter-gatherer societies, right, or even agricultural revolution, so, but hunter-gatherers, so let's say you got males and females, typically speaking, with the exception of Amazons, and I don't know that story. I would love to see their adaptation and accommodation. So typically, males would be far more likely to go out and have a series of single focus activities, right? Which predisposes them for very single focus activities, and they're super good at adapting or accommodating out extraneous information. So they would, for instance, go out and hunt the wildebeest and kill the wildebeest and drag the wildebeest back home, feed themselves first, feed their family second, have sex, go to sleep, do the same damn thing the next day. So if you were not a wildebeest, somebody to have sex with, or somebody who could kill them, then you were really easy to accommodate out extraneous information. Whereas women, historically speaking, were more likely to stay behind in social groups, right? And have way more children than they could keep alive. One of them's going to fall off of something, another one's going to get eaten by something, right? So women, as far as the bell curves go, it's a bimodal distribution. Women are more likely to be able to do multiple focus activity, right? And a little harder time adapting or accommodating out information, right? Let's just take a poll. How many of you guys in here can have a conversation and listen to another conversation at the same time? Ooh, 2, 3 guys. We didn't have any guys in the last one. So you can have a conversation and listen to it. That what you're doing there is you're basically multiple focused, right? So typically speaking, and this is not for every male or every female, males tend to be far more likely to be good at single focus activity and really good at extraneous information being just thrown out. And women tend to be much worse at that. They're good at multiple focus, but they're also a little harder time actually adapting or accommodating out information. So let's say you want to get romantic with your wife, but the sink is dripping, the kids are all dirty little smatchets, and your mother-in-law is coming over in 20 minutes. You're going to get single focus activity? Probably not, right? However, it's typically a little easier for males to adapt out that extraneous information, right? And so you've got these bimodal distributions of those kinds of things. Okay, so that's adaptation or accommodation. And this is not for every male or female. This is just predispositions for populations, right? There are males that fall on one end and the other, and females that do too. All right, so go ahead, Charles.
So this is comic relief. Have you ever seen the video about a man that explains women's brains versus men's brains?
No, I have not.
I will show you after class.
Okay. Are you sure you want to watch it with me, or you just want to like send it to me?
I showed my mom, so we should be okay. And my grandma, she approved.
Your grandma approved. So my joke about that is always like, if a woman wants a male's like exclusive attention, you just have to be the wildebeest, right? You got to be the wildebeest, and remember that the things that you will magically, without your conscious mind wanting to, attend to is sex, fear, and food, right? And you can do all three if you want, if that's your thing. I don't care. Okay, so we are moving. It's people, you know, on the internet, you'll figure out that people like every damn thing, ever. Okay, so we're going to move to muscle physiology, and you guys are doing muscle physiology next week in lab. Okay, you guys are having your lab practical this week in lab, although it's not very practical, it's really lab exam. How are you doing on your lab exam? Don't know? Okay, what?
It doesn't like connect to the class at all.
Well, it will next week.
Okay.
And they do endocrine far earlier than I get to endocrine. You did connect with like cell transport. You connected with that, and you're going to connect here. So you'll connect more than you ever could, or ever did before, but we're working on it. I can't get to endocrine this quickly. I wrote that endocrine lab, though, so if you hated it, you can tell me. I thought it was actually pretty good. I liked it. Like,
was that the new one we did last week?
Yeah, I liked it. What?
The frog.
No, the rat.
Or the rat.
Yeah.
The rat.
There
Okay.
was- I wanted like a flow chart instead of having to find the information in like text.
Send me that suggestion so I can fix it for next time, because it was like the first one. And I don't teach labs, so I hadn't, they were like, did you do it? And I was like, well, I did it by myself in my house, and I did great. I got all the answers right. So, I did the best I could without teaching lab. So I just had to send it out there and like, here's my baby. And they're like, oh, I hated this part. And I'm like, it's fine. I did send it to you 5 times before for revisions. And you didn't say a damn thing. So now you can shut your pie hole. But no, I will take revisions. Those suggestions would be good. And I can even have like a flow chart in which you fill in some parts, right? Like it's like got part of it filled in and then you fill in the rest. But that will be really useful to you when I get to endocrine, because it's almost the same way that I teach endocrine. So it's just going to, and then next week you guys are doing muscles. I'll be doing muscles when you guys are doing muscles. The muscle lab is fun and it's good. And if you really want to learn it, volunteer to be the one that gets zapped, right? Because it doesn't even, well, it hurts a little, but let's just be serious. But it only hurts enough that you won't forget it. You'll be like, oh, I totally remember because I got zapped. So the muscle lab. So we are going to start on muscle today. And please remember, is there anybody in here who did not go all the way down to the sarcomere level in anatomy?
I'm in anatomy.
Because you're in anatomy right now. But we did go down to the sarcomere level in anatomy. Yeah? Oh, you haven't gotten there yet? I have. Okay. So you'll have a little bit of a hard time, but you're bright, so you'll be fine. We are going to go all the way down to the sarcomere level. I'm going to assume that you have already done that, meaning that I'm not going to spend as much time building the parts that I know you learned in anatomy, because it gets a lot more complicated than that. So I'm going to start from there, go through that part really quickly, and then add a lot of detail. This stuff is really interesting. It's complicated, but interesting. Okay? So it's not like a Gprotein in which it's complicated and you're like, I don't even know why I need to learn this. It's complicated and interesting. So what you need to know before you come to class on Monday, all those damn sarco and myo words, go make sure that you can use them. Sarcoplasmic reticulum, sarcomere, sarcolemma, sarcoplasm, myofibril, myofilament, all that crap. Make sure you remember it, okay? Because I'm going to assume that you did. So if you are having trouble with it, interactive physiology is really good for reviewing the anatomy, okay? The interactive physiology is great. Okay, So let's just start talking about the functions of skeletal muscle, and then we'll get into the details. So skeletal muscle, you know it makes you wiggle, but that's not all it does. It's also responsible for postural stability. I need skeletal muscle in order to hold myself up. It's also sometimes responsible for holding things in, right? These skeletal muscles right here in the perineum hold junk up. What happens to those muscles if you have a whole bunch of big-headed babies? Yeah, and then what happens to your uterus? Where does it end up? Hanging out down here, floppy floppy. Uterine prolapse. So it's good birth control. You know that. Okay? Whole bunch of big-headed babies. I teach the 17-year-olds, the seniors, every other semester, and don't think I don't tell them that. I'm like, they're like, oh my God, that's worse than gonorrhea. Floppy uterus hanging out. So also, your skeletal muscle sphincters guard your openings, right? Bottom of your digestive tract, the bottom of your urinary tract, keep you from peeing and pooping down your leg. Heat production. What you are doing with skeletal muscle contraction, we will see, is taking chemical energy in the form of ATP, converting it to mechanical energy. And since energy conversion is really inefficient, you always generate a lot of what as a byproduct? Heat as a byproduct. That's the reason that shivering works, to heat up your body. Skeletal muscle also stabilizes your joint. So if you strap a strong skeletal muscle across a weak joint, it helps to stabilize the joint. And then skeletal muscle also helps keep your fluids moving. So we know that the heart is the primary reason why blood moves through arteries. It's actually not the primary reason that blood moves through veins. Blood moves through veins in large part due to skeletal muscle contraction around the veins. And of course, the lymphatic system doesn't have a heart at all, right? So why does stuff move through the lymphatic system? Well, you squish on the vessels, primarily with skeletal muscle. I mean, there's smooth muscle on the wall too, but primarily skeletal muscle. Okay, so here we go. We are going to start with skeletal muscle. First thing I want you to understand about skeletal muscle is it's highly, highly innervated. What do I mean by that term, innervated? Lots of neurons attaching to skeletal muscle. It's also highly, highly vascular. What does that mean? Lots of blood. So let's look at how it's innervated and let's look at how it's vascular. Ready? Okay. This is a histology image of skeletal muscle showing you the actual axon and the axon terminals, the telodendria, splitting and going to one, two, 3, 4, 5, 6 different cells. Now you can't see it attaching to all 6 different cells, but here's something I want you guys to understand. Every single one of these cells needs individual, personal attention from the nervous system or else it's paralyzed. That means that if this fiber, this axon telodendria, ever gets damaged, this cell right here is paralyzed unless and until you get reinnervation. So that means that this is an incredibly, incredibly innervated tissue. You have to talk directly to every single cell. And that's pretty damn impressive when you think about the fact that you may, in your gluteus maximus, have 100,000 muscle cells. Does that make sense? So this is important, and it also lets you understand a little bit about nerve damage. Have you ever known that nerve damage can result in muscle paralysis, nothing, or muscle weakness? Right? Can you understand how if I damage these two, it might result in muscle weakness, but if I damaged up here, might get complete paralysis, right? And by the way, the peripheral nervous system, one of the things that we will learn is the peripheral nervous system has the possibility, but not the guarantee of reinnervation, so you may actually regain some functionality over time. If it's the CNS, you're screwed. You're not getting any reinnervation. Okay, so this is highly, highly innervated. So, question for you. How does cardiac muscle get around the need for individual innervation of every cell? Connected with what? Intercalated disc. What's in there that allows for communication? Gap junctions, yeah. Smooth muscle is also connected by gap junctions. Skeletal muscle doesn't have any gap junctions between them. Okay. In addition, cardiac muscle also has spontaneous contractions, right? It has pacemaker cells. Smooth muscle does in some, and skeletal muscle never does. So you have to talk to each individual cell, or else skeletal muscle does nothing. So it's like they always have to be told exactly what to do. I think about this like my teenager, my especially 18-year-old, right? So this is Gavin right here, my 18-year-old. If you tell Gavin to take a shower Or in our house, we ask and not tell. If we ask Gavin to take a shower, we don't tell anybody anything, because I don't like being told what to do you? So we ask Gavin to take a shower. However, if we also do not ask Gavin to wash his hair, he ain't doing it. If you do not ask Gavin to put on clean clothes after he takes a shower, he's going to put on the same dirty underwear that he wore before. He totally will, right?
He's 18 and acts like this?
Oh my God. I think he's going to get killed by his roommate, honestly, when he goes off to college, right? And it is always like, you didn't tell me to do that, right? Every single time since he was 3. You ask him to take out the garbage, and he will not put the liner back in the garbage can because I did not tell him to do that. So here's what I did on his mirror. Take a shower, wash your hair, wash your body with soap, put on clean clothes, put your dirty clothes in the laundry. So he's a skeletal muscle fiber. That's all I'm saying. Okay. I love him dearly. I think his roommate's going to kill him. His first roommate, aren't they going to kill him? Pretty sure they are. Okay. Huh?
Or just kick him out.
Just kick him. Yeah, he can't get kicked out of a dorm. He's going off to college.
He'll just be single forever.
He will be single. It's probably good. I mean, he knows how to do it. It's just that he's always expecting somebody else to give him the impetus for doing those things. And he's like,
the sweetest girl ever, and she'll
and then she's going to leave him.
she'll wash his hair.
She'll wash his hair.
He's going to need someone to tell him to do those things, though. So
I have tried. I'm just like, I don't even know why anybody would put on dirty underwear ever in the history of ever. Right? It just shouldn't happen. Okay, so back away from Gavin. Back from Gavin. Okay, let me tell you a little bit of terminology that you're going to need to make sure that you remember. This right here, you have enough information somewhere in the resources of your brain to know that this is a somatic motor neuron that is responsible for innervating skeletal muscle. Does that sound vaguely familiar to you? It is a somatic motor neuron. So a motor neuron from the somatic division of which one? The CNS or PNS? PNS, right? Okay, so we are eventually going to put all of that information in with physiology. We're going to put the A together with the P. And you already know the A. I'm just going to start using it as if you were familiar with it, okay? And then we'll all get used to it again. Okay, the other thing is that each individual little skeletal muscle fiber, so we're going to use a different staining technique right here. This is stained to look at the capillaries instead of just the muscle fibers. Each individual little skeletal muscle fiber is, this is a supravascular tissue. Every single muscle fiber has a little snuggly net of capillaries all the way around it. So this is incredibly vascular tissue because it's super energetically expensive. And we'll see how it uses all that ATP. So if you look at this and you imagine, okay, this is a cow muscle, not a human muscle, because we don't want to be super creepy. And we take a cross section of it and we sit it out on the counter, right? What color is it? It's red. It's a steak. It's red. It's a cross section of a cow skeletal muscle. Part of the reason that it is red is because there are capillaries snuggling around every single cell. Does that make sense? Hemoglobin in the capillaries snuggling around every single cell. Okay, so it's supravascular tissue, highly innervated tissue. Okay, now let us dig into the inside. Okay, so let's acknowledge something that's super annoying. Some Yahoo decided that all of the terminology that you spent your entire 1610 learning needed to have new names for all of the cell parts when you learned a skeletal muscle. So instead of calling it a cell membrane, now you have to call it sarcolemma. Instead of calling it cytoplasm, oh, it's sarcoplasm. Instead of calling it the smooth endoplasmic reticulum, it's the sarcoplasmic reticulum. So they renamed every damn thing inside of it. Go ahead, Yuenta.
with those little capillaries that break when you get a bruise?
Yes, among other things. Yes, they break, but usually what you're seeing is you don't usually get you can get muscle bruises. You usually get them in the connective tissue in your skin, right? But you have had a muscle bruise before. Have you ever really smashed into a muscle and had it bruised? So with that, you would actually have capillaries broken in the muscle, right? It doesn't usually get that deep though.
And do you see that?
No.
Okay.
It would usually just be really painful and inflamed. It wouldn't come to the surface necessarily. It would be carried away.
Too deep.
Yeah, too deep, right? Okay, all right, so they renamed everything, and I'm really sorry, and I wouldn't make you learn the terms except that that's the terms that they use and the ones that are in every textbook. So a skeletal muscle fiber is a cell, but but it doesn't look a darn thing like any of the cells you looked at in 1610, does it? Okay. In addition, they can be up to a foot long, but they're microscopically thin. And they got a whole bunch of things inside of them that no other kind of cell will need, because these things, these Vienna sausage looking things, are related to the special function of contraction. Okay, so before we get to those, let's talk about the stuff that it has in common with other cells. Okay, of course it has a cell membrane, yes? And the cell membrane is just like cell membranes are. It's made of what? Phospholipid bilayer. They just renamed it because they're jackasses. They call it now the sarcolemma. Okay, the cell membrane of a skeletal muscle fiber is called the sarcolemma. And the sarcolemma has some new functionality or new structures that will give it new functions. This one's going to take a second to explain to you, and you'll start to see how it's important later. Okay? See these little green things right here? They look like this from the top. They look like a pore. And sometimes you learn them in anatomy and sometimes you don't. I don't go into that detail in anatomy because I know I'm going to do it in phys. These are called T-tubules or transverse tubules. So let me show you what they really look like. Don't get freaked out by this figure yet because we're not going to learn all the details of it. Okay, a T-tubule is a folding in of the cell membrane that allows you to take an action potential from the surface to the deeper portions of the cell. And by folding in, it's a tunnel into the deeper portions of the cell. They call it an invagination, as in the word vagina, right? You're not going to forget that, are you? T-tubule, right? Folding into the cell. Here's what it looks like, right? So we will get to this detail in just a little while. So an action potential doesn't have to stay on the surface. It actually can go deep into the cell. That will be important because the action potential getting into the inside of the cell which is what will cause contraction. Okay? So that's a T-tubule or a transverse tubule. Everybody okay with that? Okay? So what it looks like on the outside is this. Okay? So what else is in there? The sarcoplasm, now there isn't a whole hell of a lot of sarcoplasm because the thing is really full, but there is some sarcoplasm in there. It's just the intracellular fluid or cytoplasm of the cell. What's in there? Some stuff that other cells have and some stuff that other cells don't. First of all, do you remember that skeletal muscle fibers are multinucleated and that their nuclei are peripherally located? This one's only showing you one, but they are multinucleated. Can you see now why the nuclei get pushed up against the periphery? Because the center's kind of busy, right? There's a bunch of crap in there. So they're multinucleated, they're peripherally located. Got it? Okay. As far as mitochondria, which are represented in pink here, there are boatloads of mitochondria compared to other cells. Does that make sense? Okay. Because it's really energetically expensive tissue. And what are mitochondria for? Cellular respiration. I hate you, Melanie. Okay, so it's fine. So a whole bunch of mitochondria. And then two things that are not drawn on here but are an important part of this story. You ready? In here, in the sarcoplasm, not drawn but important, are two things that are storing stuff. One of them is that individual skeletal muscle fiber has a little piggy bank of glycogen. Okay, it doesn't share with other cells, and it's not like a savings account like the liver is. The liver is a savings account, right, that it can use for the rest of the body. This is a piggy bank, and it does not share with others. Okay, so now we've got glycogen, and we've got mitochondria. We're starting to tell a story, aren't we? So we've got mitochondria. Okay, we've got stored glucose as glycogen. What's one other thing that we need in order to complete this story? Oxygen. And what we have also in there is oxygen stored, small quantities of it, bound to a protein called myoglobin. Myoglobin is similar to hemoglobin. Hemoglobin binds to and carries oxygen around in the bloodstream. Myoglobin binds to and stores small quantities of oxygen inside every skeletal muscle fiber. So we've got oxygen, and this is on myoglobin. So what we have is skeletal muscle fiber, even if you haven't eaten in 72 hours, can potentially get you out of the way of a bus, right? Because what can it always do? Little teeny tiny bit of cellular respiration, right? Relatively quickly. Doesn't even have to wait for the cardiovascular system and the respiratory system to catch up, does it? Because it's got it all stored internally. Does that make sense to you guys? So you leave your skeletal muscle fibers there. And to reinforce this, one of the reasons that, so you probably noticed that after you work out, your cardiovascular and respiratory rate do not immediately go back down, right? And you think, well, you just can't turn them off that quickly. Yeah, you can. One of the reasons that they don't immediately go down is the last thing that you have to do after you finish working out is you have to repay that debt to all of your skeletal muscle fibers, all of them that you used, right? And you need to repay the glucose, even if you have to break down glycogen from the liver in order to do it. And you have to repay the oxygen. They call it repaying the oxygen debt, but it's really the oxygen and nutrient debt, right? So your metabolism actually stays up even after you stop working out because you never want to leave your skeletal muscle fibers in the situation in which a bus is coming at you or a saber-toothed tiger and you can't even get a little bit of energy to get away from it, right? So the last thing that you did, the last energetically expensive thing that you did was to replace that. Does that make sense to you guys? Okay, so the other thing about myoglobin is when oxygen is bound to myoglobin, it makes the cells red. So now we have two reasons why your steak is red. Your steak is red because oxygen is bound to hemoglobin, and also because oxygen is bound to myoglobin. Yeah? And if you set your steak out on the counter for a little while, the oxygen starts to dissipate off, and what happens to the color? It gets brown. And if you want that to happen more quickly, in addition to cross-linking the proteins, you just cook it, right? And then the oxygen dissipates off really quickly. Is that okay? All right, so the last thing is to address the elephant in the room. And the elephant in the room is this big Vienna sausage-looking thing. It's called a myofibril. Here's what a myofibril is generally before we get into the specifics. The myofibril is a big cable-like bundle of contractile proteins attached to the cytoskeleton of the cell, right? So when you get that myofibril to contract and it pulls on the cytoskeleton, what happens to the length of the cell? It shrinks, exactly. Now, just to put it simply together with what you know from anatomy about muscle contraction, how can I make my facial muscles move? Well, what I do is I stimulate enough myofibrils to pull the entire cell. And then if I stimulate enough cells, I can move a whole fascicle. If I stimulate enough fascicles, I can move a whole muscle. And if that actually generates enough tension to move skin, then I can do this. If it actually takes more tension to move a bone, and even more if the bone actually has something on the other end, right? And that all has to do with how many contractile proteins you're actually contracting, okay? So that's a myofibril. Now, before we go any further, I want to talk about how you make your muscles stronger generally before we get into it specifically. So, I want to imagine that I start working out, okay? And what I'm going to do is, let's say you're doing biceps curls, right? And if what I do is I try to lift something with my biceps brachii, my brachialis, over and over again, that's almost at the limits of my capacity. I don't know what I could lift with my bicep, maybe 30 pounds? Or, I don't know. I do push-ups every day. I hope I can lift more than 30 pounds. But it would be different for me than him. He's got more testosterone than me. So what I will do is I'll try to pick up something 8 times, which is almost at the limit of my capacity. That make sense? And what I'm going to do is I'm communicating to my muscles when I try to do that. And what I'm telling them is that you are not strong enough for the task at hand. Yes? Okay? And when I do that, if I, the next day, will rest and give myself some proteins and some ATP, I will make more myofibrils inside that cell. And more myofibrils will give what kind of contraction? Stronger or weaker? Stronger contraction. Does that make sense? Okay, so you can get more myofibrils. Here's what you don't get. You don't get any more muscle cells. You were born without all the muscle cells you're ever going to have, but you can certainly take each muscle cell and give more contractile proteins on the inside. But you don't want to do that thing that people do, which is you work out but you don't feed yourself, right? Because can you do any protein synthesis if you don't have adequate proteins in your diet? So in order to build muscle mass, you need to eat what? Protein. And in order to build muscle mass, which is a what? Catabolic or anabolic process? Anabolic. You also need energy in order to do it. And the other thing that people do is they will work out their biceps, and then the next day they will work out their biceps. Any idea why that wouldn't work as well? Because the next day, instead of using that energy for protein synthesis, I'm using it for muscle contraction. That makes sense? Okay? So what you need to do when you're trying to build muscle mass is you need to communicate and then give it some time to respond. Communicate, give it some time to respond. That makes sense? Now, why would it work different in the two of us? Because testosterone increases protein synthesis in skeletal muscles. So if we were the same size and had the same strength and did the same activity, who would get a more dramatic response from it? He would. Testosterone causes protein synthesis in skeletal muscles far more than estrogen does, right? So you can see a dramatic increase. And now you can see why people shoot up testosterone too, right? Because you're trying to get all big and buff. That make sense to you guys? Okay, so let's break down this myofibril and talk about it. Okay. Oh, I forgot to tell you one more thing. The myofibrils are wrapped in, do you see this like net that looks, it's not a capillary net because that would be on the outside of the cell. On the inside of the cell wrapped around each one is what we call the sarcoplasmic reticulum, which is the smooth ER in skeletal muscle fibers. Does anybody remember what the smooth ER does in most cells? Hardly anybody ever remembers that. What?
Transports stuff.
It does in transport, but you're thinking mostly Golgi. What? Not ribosomes. Nobody ever. Lipids, yes, lipid synthesis.
I thought it dealt with toxins too, poisons.
It can, because that's part of its function, but its primary function is lipid synthesis. But in addition to that function, this one in skeletal muscle does all of that stuff, plus stores calcium. Okay, so keep calcium in your mind, because calcium is going to be stored right here next to the T-tubules. These are actually called the lateral sacs of the sarcoplasmic reticulum. Okay, so that won't be important until later, but keep it in your mind. Okay, so now let's break down a myofibril and look at what's inside of it. You guys have done this before, okay? If you look at a myofibril, notice that what you're going to start to see is this repeating pattern, right? Jagged lines and stuff in the middle, right? And you guys learned that in anatomy. You learned that from one jagged line to the other jagged line is called a what? Sarcomere, exactly. So the relationship of sarcomeres to myofibrils in my mind is like boxcars to a train, right? Sarcomere, 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 line up, make a myofibril, attach the myofibril to the side of the skeleton, right? Okay, that makes sense? So what we're going to do is we're going to take one of those and we're going to look at its structure. We're going to break it down that way. I'm going to draw it 2D, but please note that it actually is a 3D structure. And we're going to start with the ones that you learned already in anatomy. And then next time we're going to learn a lot more than that. Okay? So let's start with the simple parts. What a myofibril is, it's basically a bundle of little skinny proteins that interact with one another. They're contractile proteins. So here we go. We've got jagged line to jagged line. The whole thing is called a sarcomere. Okay? The jagged line is called a Z-line. It's the boundary of the sarcomere. In your anatomy book, they call it a Z-disc. It's the same thing. Okay? And then we've got two really important proteins and then some other proteins that we'll learn about. You didn't probably learn about most of them in anatomy. Okay, so what we have, if we have some proteins that are attached to the Z-lines and extend toward the middle. These you should remember, they are the thinner of the two contractile proteins, so they're called actin, thin. Sometimes they put myo and sometimes they don't. Filaments, they are a protein, they would have been made inside the cell on the ribosomes, right? Okay. Actin is the name of the protein, thin because they're the thinnest of the two. Myo means muscle, filament means long skinny thing. Okay? And then the other protein, the other important one, and there are some other proteins too, is this big thick one that's in the middle. And this one is myosin. And it is the thicker of the two, and it's also a myofilament. Okay, so preview, and we will get into muscle contraction in much more detail than they do in anatomy, but I want to make sure that I put it together with the stuff that you learned in anatomy. Okay, so what we are going to do is we're going to get a preview of muscle contraction. And first thing is I just need to add a little bit more detail to this. Actin actually has binding sites on it. They don't look like handles because they don't project that this is the best metaphor that I can come up with. So these guys are on actin, and they're actually just chemical binding sites for myosin. Those are called the actin active sites. So keep those in mind for just a second. And then the myosin ones you can actually see. So let me show you. Myosin ones actually do project up. So here's what myosin looks like. It has these little projecty, sticky uppy things here. Okay, these are called myosin heads. And I think of them as hands that can reach up and grab the handles. Okay, so simple preview, and then a little bit more complex. So how do we contract a sarcomere? Upon stimulation by a somatic motor neuron, some magic happens. It ain't magic, I just haven't explained it yet. Some magic happens, and then ultimately what will happen is the myosin heads will reach up and grab the actin active sites and pull. And then what happens to the distance between the Z lines? It decreases, right? And if I contracted enough sarcomeres, I can contract a whole myofibril. Contract enough myofibrils, I can shorten the cell. Enough cells, I can shorten the fascicle. Enough fascicles, I can pull in the tendon. Does that make sense? Okay, but note to self, it does not happen in one fell swoop like this. Okay, so what we will get is we will next time start to dig into the details about how it's going to happen, but I'm going to give you a preview of things to come. Here's the preview of things to come. First off, whenever a myosin head and an actin active site are bound to one another, it's called a cross bridge. Okay, so here is how this works to give you an appreciation for what we're going to learn next time. Okay. So I want you to imagine that monkey bars, yeah? You with me? Only I'm going to do them this way, okay? So I'm going to do monkey bars. Here's what I'm going to do. I'm going to reach up and grab and pivot, right? And then I'm going to reach up and grab the next one because I can reach it now, right? And pivot. And reach up and grab the next one and pivot. So I'm not sliding it, I'm ratcheting it. But I'm doing the same thing on the other side. So it's really like you got a big beefy kid who's moving the end of the jungle gym toward him, right? Okay, so he's standing in the middle and moving the jungle gym toward him. Now, let's talk about what this has to do with anything. So I'm going to give you an appreciation of why the hell skeletal muscle is so energetically expensive by telling you that every single time I reach up and go, that costs you an ATP.
Oh, gee.
My thoughts exactly. So I've got a single sarcomere, and remember that a sarcomere, although I'm showing it 2D, it's actually 3D. Yeah? Just one sarcomere, right? We're going, but also 3D. And how many sarcomeres are in a myofibril? I don't want to count them. A bunch. How many myofibrils in a cell? A bunch. Yeah? How many cells? Up to 100,000, right? So this is incredibly energetically expensive tissue, right? And the other thing is that after you have this tissue, it never completely relaxes. It always maintains partial tone by at least a little bit of contraction. So if you've ever slept with anybody who's super hot, poke them. They're still hot when they're sleeping because they got partial muscle tone, right? Okay? So why is somebody who's really muscular, why can they eat like 7 times the cheeseburgers you can? Because it's all of this incredibly energetically expensive tissue that they have that once you generate it, it never is completely relaxed, right? So you can increase your metabolism dramatically by increasing the amount of muscle tissue that you have. Does that make sense to you? Okay, so last little tidbit right here is just to acknowledge the banding pattern that you end up getting. If I use a protein staining technique, wherever I have thick, dense protein, it's going to be darker. Wherever I don't, it's going to be lighter. So let's just acknowledge what you saw before in anatomy. Myosin is thicker than actin. So this band is going to be what? Dark or light? Dark. This is the A band. In the middle right here, it's slightly lighter than dark, though, right? Because it doesn't have overlapping proteins. Anybody know what this is called? H zone. Okay, it's dark, but it's not as dark, right? And last thing, from the end of this A band to the beginning of the next one is going to be what? Dark or light? Yeah. This is light. This is the I band. Okay, last thing, and I'll let you guys go. So, if this is how it actually works, and I contract this sarcomere, tell me which of the bands will not change length. A band stays in the middle because it's the fat kid pulling it all in. Strong kid, I guess. Strong kid pulling it all in, right? What about the H zone? It's going to shrink, might even completely disappear if you contract maximally. And the I bands? They're going to shrink as well. That makes sense? So as you guys are packing up, when I learned this in phys lab, we actually used to still use animals in phys lab. We would take a frog, gastrocnemius, pit the frog so that he couldn't feel anything, stretches gastrocnemius across a light microscope and zap it with an electrode. What we would see is watch his tissue go from being striped to being dark. Relax again, striped, dark, right? Poor frog, but I have never forgotten the lesson, right? Make sense? Okay, I will see you guys on Monday. No quiz until next Wednesday. What? Aside from the one you have due at 6, don't forget it. And don't send me emails at 6:01. I'll delete them. What do you have to show me, Nicole?
Okay, that's funny. It is funny. I show this to everybody that's like, what?